So good morning, everyone. Dear students, how are you doing? I believe you are coping well. I pray that everything will go well with you. So please, I want just one person to respond to let me know you are actually hearing me.
Yes, ma'am, we are here, ma'am.
Okay, thank you so very much. God bless you. So we are going to start our lecture now. Let me just share my screen so we can start. All right, so we want to start with microbial variation and heredity. That is actually our last lecture on my path. I showed you the lecture scheme. I showed you the lecture scheme at the onset of this lecture. So this is my last topic, but it's a bit large. We may not be able to complete it in 1 hour. So we might have to take the last lecture next week, Friday.
Ma'am, we want to hear you, ma'am.
Okay, can you hear me now? I had internet.
Yes, we can hear you.
And you can see the screen now.
Yes.
All right, thank you so very much. So we'll now start the lecture. Please, if I get logged out again, just be a bit patient. I will log in again. I'm actually using 2 internet connections to make sure that everything goes well. All right, so we're going to start with microbial variation and heredity. We take the last lecture next week, Friday, by the grace of God. So on Tuesday, if you know you are scheduled for practical, please ensure to attend, because if you start to avoid practical, that is the beginning of failure, because your entire practical serves as your continuous assessment and is a very chunk part of your mark at the end of. Thank you. So, excuse me. All right, so we have microbial variation. Let's do some introduction. In this session, we are going to be looking at the subject of how microorganisms pass down genes that determine traits to their offspring. You know, even in human beings, in plants, and in animals, this subject of heredity and variation also exists. So, but we are microbiologists and we want to talk microorganisms pass down traits from parents to their offsprings or to their progeny. So we are going to try to understand heredity, the importance, because it helps to
We can't hear you, ma.
I never did. I said, guy, chief.
All right, so we're starting all over again. Hopefully it will work well. I've changed the source of internet again today. So traits passed over time can change a species. With time, some traits do change in species. This change in traits enable organisms to adapt to specific environments for survival. So traits can change, and sometimes traits do change just for the purpose of adaptation and survival. So let's look at briefly some definition of terms which you should pay close attention to, since we are going to be having CBT exams most of the time. What is the problem? Somebody came and collected now. Who is the person? Excuse me, I have a class. You are disrupting my class. Please call your colleague, because one of my colleagues, I was having a lecture, they came in and I gave them the books. They find out who is the person. I'm very sorry, my dear students. So definition of terms, heredity. What is heredity? Heredity is biological inheritance. It is the likeness of the characteristics of progeny, that is offspring, and their parents. For example, look at you, look at me. What are those characters that I inherited from my parents, from my dad, from my mom? What did you inherit from your dad, from your mom? Sometimes you have a fair in complexion mom, a dark in complexion dad, but you are light in complexion. So you might have a fair in complexion color, but your height might be that of your dad or your mom. If your dad is very tall, even though you took the complexion, you take the complexion of your mom, light in complexion, you might be as tall as your dad. So, you know, that is what heredity is all about. But we also have variation. That is the differences between offsprings and their parents. Variation refers to change in an organism relative to its parent. And that is why sometimes parents will be like, where did you get this from? I am not like this. I am not like this. But you know, they have forgotten that characteristics in great-grandparents can come and show up in the life of the offspring, and the two direct parents might not have that trait. So if you might have a trait that your great-grandfather has. It might come out as a recessive character later on in life. We also have heritable variations brought about by changes in genetic substances. That is, genotypic variation can occur as a result of changes in the gene, either through mutation, loss or acquisition of new genetic elements. They have started noise again. All right. So non-heritable variation. Changes or variation brought about by environmental change. This kind of variation are not permanent and are basically phenotypic. Let me use human being to explain so that you will understand what is going on for even microorganisms. If, for example, somebody comes from a wealthy family, is well fed and looks healthy, God forbid, and something happened, maybe kidnappers kidnapped, and for months the person was not well fed, or the parents passed on and there was nobody to give adequate care, or there was war or something, and then the person has to start feeding poorly. The physical change, which is the phenotypic changes, will be different from that chubby, well-fed person. But that change arise because of the prevailing condition and the environmental condition around that person at that time. If the person is brought back to normal condition, it will change again and start to look healthy. The same thing with microorganisms. So we also have phenotype. Phenotype is the characteristics expressed by a cell at a given time. Organisms can show red color, brown color, it may be wrinkled, it may be round, it may have any shape. The same thing, a dark parent and a fair complexion parent come together, but the child decides to be fair or black. That means the person phenotypically expressed dark coloration. We have phenotypic changes or phenotypic variation. For example, in microorganisms, under uniform condition, when the conditions are right, bacteria possess constant characteristics for all the cells. So it makes bacterial identification and classification possible because they look alike. You know, okay, if I see this cell, this color, it is round in shape, I know this is a coccus. If it is straight chain, I know it is streptococcus. If it is clumping together, I know it is staphylococcus. When isolated, pure culture stored in the laboratory under the same proper condition remain the same. But if you study the same organism under different stages of growth or under different environmental conditions, changes in appearance will definitely take place. And changes in metabolic activity may be detected. All these are phenotypic changes imposed on the cells of the bacteria by the environment. This is known as variation or physiological variation. Another definition we want to look is genetics. Genetics is the science of heredity and variability of characteristics of an organism. What is a genome? A genome refers to the entire gene possessed by a cell. Microbial genome encompasses all chromosome and extrachromosomal genetic material. Extrachromosomal genetic material is also known as plastic. Genotype. Genotype is the genetic constitution of a cell. That is, what is specific about the DNA for that particular individual or that particular microorganism or cell. Genomics. Genomic is the study of genomes as an entity. What is a chromosome? A chromosome is a thread-like structure of DNA, and that is nucleic acid and protein that carries genes. Plasmid is a small DNA molecule within a cell that is physically separated from a chromosomal DNA and can replicate on its own independently. It is also known as extrachromosomal DNA, capable of independent replication. And you can also see something like F factor. F factor stands for fertility factor. And this allows genes to be transferred from one bacterium carrying the factor to another bacterium that lacks the fertility factor during conjugation. And of course, we have the high frequency recombination cell. It is a bacterial cell with F plasmid, that is fertility plasmid, integrated to its chromosome. Let's look at some few examples of phenotypic variations. Morphological changes of variation. Morphology is the branch of biology that deals with the study of structure and forms of living organisms. And I explained earlier, when a bacterium is inoculated into a fresh medium, it undergoes different phases of growth. At the end of lag phase, the organism may appear large, the cells may appear large. As it proceeds to exponential phase, and then the stationary phase, the cells may appear smaller, but they will be uniform in size because of cell division. You could also have cultural changes or variation. For example, the pigmentation may change. The pigment of a culture of bacteria may change. due to several reasons. For example, temperature. Temperature do change the pigmentation of some organisms. I gave you an example of Serratia marcescens that usually will produce a distinct brick red pigment when incubated at a room temperature. But if you place that same organism in the incubated and you increase the temperature to 37 degrees centigrade, then it will lose its pigmentation. You will no longer see that dark brick red coloration. So organisms can change. I have cultured an organism with a specific media before, and it was cream colored. But when I transferred it to 37 degrees centigrade, it changed to orange color. And I was almost not able to recognize that this was my organism until I took that orange color and then incubate it back and place at a different temperature, the initial temperature, and it gave me back the initial color of cream. So the kind of agar you use, the temperature can also alter pigmentation in microorganisms. The shape and structure of organisms can also change. For example, there can be colonial changes. Sometimes when you grow microorganism in a certain substrate, it will give you different shape. If an organism is getting older in a specific substrate, it might become wrinkled, whereas the cell was initially smooth. The example I gave you here, if you grow an organism in a substrate that have disaccharide, the organism will produce an enzyme. It will produce an enzyme that can only be stimulated by this disaccharide sucrose. Smooth colony may also change to rough colony, like I explained, when the environment is unfavorable. Maybe the nutrients has reduced, and then the organism is no longer able to cope. It begins to wrinkle, just like in human being. You are born, then towards your teenage age, you become very beautiful. As you approach 25 to 35, your skin glows, you become beautiful. As you approach 45, 55, you begin to get dark in complexion, and by the time you are 60, 65, 70, the skin begins to wrinkle. So also with microorganisms. You can also have physiological or biochemical variation or changes. Usually, when you talk in terms of physiological, like resistance of microorganisms to harmful chemicals, maybe like Dettol or any other chemical, young cells are more susceptible because the older cells somehow may have been adapted to these disinfectants or whatever chemicals. The type of enzyme a particular species of microorganism will produce, like I said earlier, depend on the substrates. For example, if you grow E. coli in an environment or in substrate that has beta-galactose, then the beta-galactose taken in by the E. coli will force the E. coli to produce enzyme that will be able to enable it to digest the beta-galactose. Hence, it will produce the enzyme beta-galactosidase, because that is the enzyme that will split or break down the galactose. Otherwise, the organism will die of hunger. If the organism cannot produce the enzyme that will help it to break down the sugars, then it will not be able to survive. So to be able to survive, the substrate will induce the production of this enzyme, and then the enzyme will be used to split or break down the sugar, for example, galactose. And in terms of genotypic, in terms of genotype or phenotype, it only means if the organism is able to secrete or produce that enzyme, it only means that that enzyme is already inherent in the genotype of that organism. It is there in the organism. The genotype to produce that galactosidase enzyme is already inherent in the organism. But without the substrate, the organism cannot express it. So the expression, to express it out, is this phenotype, because you will now see it. So what is inherent in the gene can be expressed when it is induced by substrate. Genotypic changes. Organisms possess structures called genes, which determine their characteristics. Any gene is capable of changing or mutating to a different form. Let me use human being as an example so that you will be able to understand what is also happening in the microorganism. For example, men and women, especially women, age over 40, go for tests. You want to check to be sure you don't have cervical cancer. How do they do that? There are several methods, but the latest method is using PCR that will enable them to detect the gene that can be responsible for that cancer. Usually when they know when you are born, those genes are okay. As you are getting older, and depending on the environment, for example, if you are exposed to chemical, if you are exposed to UV and other dangerous materials, the cells can begin to mutate. That is, they will change. And when they change, if you look at PCR, using PCR, you will see the gene is already changing, is already mutating. And they will be able to detect that this gene is already mutating, and in the next 10 or 15 years, it can become cancerous. So what they do most of the time is to cut a part and really check, is it going to produce this cancerous cell in the next few years because of the mutation that is taking place? So also for microorganisms, different conditions can lead to mutation. And when the gene is mutated, then the organism is going to take a different form. It will have a different characteristic. And mutation is usually a stable heritable change of bacterial gene. When mutation takes place, it doesn't go back to that normal again. It remains, it is stable, unlike phenotypic changes that when the condition of the environment comes back to normal, the person also comes back to normal. But for mutation of gene, it is stable, it is heritable. So when susceptible bacteria are exposed to, for example, danger, e.g. phage, bacteriophage, which you have learned in your secondary school, some of the cells survive, but they are not going to be the same. Their descendants become resistant to phage because they already mutated. Their cells are already mutated. So what is the difference between phenotypic and genotypic changes? Like we have discussed, you will discover that for phenotypic changes, most of the cells are involved because you will see the physical change. You will be able to see the physical changes. So it involves most of the cells. For example, if somebody is going to look kwashiorkor, you cannot say it's only the hand that will look that way. You discover that from the head downward, the person will become thin. Okay? So, but for genotypic change, it might involve only a few parts of the cells, just like in human being. The genotypic changes might take place only in the uterus. Maybe the mutation is taking place only in the uterus. So the person might be developing only cancer of the uterus, for example. And then it may spread to other parts of the body. But phenotypic changes are more stable than genotypic changes. And phenotypic changes are less stable, while genotypic changes are stable. Phenotypic changes involve most of the cells. Genotypic changes occur at only the level of a single cell in microorganisms. Phenotypic change is less stable, like I said, because when the environmental condition return to normal, there's a return to original phenotype character. So phenotypic change, we can conclude it is not inherited, while genotypic change can be inherited. And that is why you are saying if somebody have a parent that has cancer or diabetes, there's likelihood that the person can develop it. So the person should be able to take precaution early enough, because it is a genetic matter. It can be inherited. But the fact that your father and mother are short does not mean you will be short if you are well fed. So the gene and the DNA. The DNA is called deoxyribonucleic acid, and it is the chemical substance that carry information which determines the characteristics of the cell and transfers this information to each daughter cells during reproduction. DNA is very essential because it is the material that helps in the transmission of genetic information from parents to their offspring. The gene is the sequence of nucleotide pairs in DNA, which code for a specific polypeptide chain. The gene lies in the chromosomes. In bacteria and many viruses, the chromosome is a single large circular DNA molecule. In viruses, is either the chromosome has RNA or DNA. Most of the time, they cannot have the two. And all bacteria have a single chromosomes per genome. So you should try to know the differences between DNA and gene. Gene are DNA stretches. That is, the gene consists of several DNA, which are encoded for different proteins. And DNA is a chemical which stores the genetic information of an organism. The gene regulates the traits, that is characteristics of an organism, while the DNA regulates the gene regulation. The gene is a small stretch of DNA, while the DNA is a long chain of polynucleotide. The genes are made up of either DNA or RNA. The DNA is made up of nucleotides. A gene is located on a chromosome. DNA is located within the nucleus of the cell. Genes are coded with heredity information. DNA encodes the genetic instructions. So this is how it is. This is the chromosome. So you can see the nucleotides inside the DNA, and you can see that the DNA is a double helix and it is found inside the chromosome. So when you are talking about plasmid, plasmid is an extra chromosomal DNA, so it will not be found inside the chromosome, it is outside. But some organisms have plasmid. So this is a bigger structure. This is what the DNA look like. And you have two kinds of nitrogenous bases, present in DNA and in RNA. Those are pyrimidine and purine bases. The pyrimidine consists of cytosine, represented by C, and thymine, represented by T. Thymine can be found only in DNA, while the RNA will consist of uracil. For the purine bases, you have the adenine and guanine, represented by G and A. Adenine always pair. That is why they call it pairs of DNA. So adenine, anywhere you see adenine, it will pair with thymine. But if it is an RNA, it will be uracil. So you can see this one is a DNA structure. If it is an RNA structure, you will not see T, you will see U because it is RNA. So in place of thymine, you will find uracil. While cytosine always pair with guanine. So anywhere you see G, you will see C on the opposite, pairing together. And anywhere you see T, you will see A. When you see A, you will see T when you have a DNA structure. And if you have an RNA structure, you will see U in place of T. So let us talk briefly about transfer of genetic material. How are materials transferred in prokaryotes like bacteria? So bacteria usually are free living haploid organisms, and they are not differentiated into gametic and somatic cells. Each cell has the potential to behave as a gamete. So in the bacteria that have, some bacteria do display sexuality. So the cells or gametes have been differentiated into donor, the male, and recipient. That is, most of the time, bacteria are free living, they are haploid. So any of the cells could behave as a gamete. They can just have cell division and then they begin to multiply. But some bacteria have also shown sexuality, where they display male and female character. Okay, so the male, there will be one of the cells that will represent male, another one will represent the female. So the male will donate, while the females will be the recipient. So we'll look at an example. So the transfer of DNA from donor cells to recipient cell, which may give rise to recombinant forms, can be accomplished by 3 mechanisms. The first one is through transformation. Transformation is the genetic alteration of a cell resulting from direct uptake or incorporation and expression of exogenous genetic material from the surrounding through the cell membrane. Transformation can occur naturally in bacteria, but it can also be artificial. So in an environment, for example, in the lagoon, in the river, in the ocean, inside the soil, on the leaf, wherever, in their natural environment, bacteria can take up DNA from another bacteria. If I have sound, for example, I can isolate the DNA. We have done that before using PCR and metagenomic tools. So when you check the DNA, you will discover that that DNA contains thousands of different bacteria and different protozoa or different viruses. So inside that soil, the DNA is freely available. So that some organisms can then pick up this DNA into their own cell. So the DNA of another type of organism can be transferred into that other cells. Bacterial transformation can be referred to as a stable genetic change. Once it incorporates that DNA, then it becomes stable genetically. But the bacteria must be competent to be able to take up DNA from the environment. That is, for transformation to take place, the cells must be competent. So what is competence? Competence is the state of being able to take up exogenous DNA from the environment. So look at this. The environment could be the soil, it could be the river, it could be anywhere. But again, it could be artificial in the lab. So let's assume that this tube that you are seeing here right now is an artificial one, or maybe it's a river. You have different DNA fragments of different organisms here. And then you have this bacterial cell that has nucleus, which contains its DNA. But if this bacteria is capable or competent of taking up DNA outside its own cell, it can do it. It can obtain the DNA. And then through cell membrane, the DNA enters into these bacterial cells. And 2 things might happen. Once it enters the bacterial cell, is either the DNA is integrated into its DNA components, or it is degraded. They are not compatible. So you may have successful or stable transformation of exogenous DNA into a cell, which becomes stable and successful. but the degraded one becomes unsuccessful transformation. So this one did not successfully acquire the DNA from the environment. If it is a plasmid, that is exogenous, extra chromosomal DNA, it's still the same process. It can be successful or not successful. The other type of transformation, and the other type of transfer of genetic material is conjugation in bacteria. Here you have the process by which one bacterium transfers genetic material to another through direct contact, that is cell to cell. These people have conjugation. They have conjugation. One bacteria serves as the donor, that is as the male. I will log you out. Victoria. Control your class, please.
Now I get it.
Victoria and Co, you are disturbing my class. All right, so the donor bacterium carries the DNA sequence called the fertility factor, that is the F factor, which allows the donor bacterium to be able to transfer the genetic material through pilus that is at home. So in the next that you can see this. This is representing the male. This is representing the female. Both have this fertility factor. So this is F negative because it is the recipient. Gumbi Yimusiba or whatever. Please, this class. I hate this. All right, so you can see that the one representing the donor is able to form what is called a pilus. That is the first thing it does. It forms a pilus or pilus, P-I-L-U-S, which shoots out like a penis. Then you have the DNA. This red part is part of the part of the DNA that it will donate from. Then the pilus connects to the cell of the recipients. And then they form a tube-like connection. And through this tube-like connection, the donor DNA replicated by rolling circle method and transfers some of its DNA to the recipients, as you can see here. Then the fragment of donor DNA is incorporated into the recipient chromosome. So the donor bacteria has successfully transferred its own genetic material to the recipient bacteria. So this is like the mechanism of bacterial conjugation. You can read more about it. Prescott you have in the library. You can read more about conjugation. This is introduction, is basic microbiology. So the third one is transduction. The third method of genetic transduction. Transduction is the transfer of bacterial genes by viruses. That is bacteriophage. We all have heard about bacteriophage in secondary school. A bacteriophage is a type of virus that affects bacteria, that infects bacteria. It is an accidental infection anyway. Bacterial genes are incorporated into a phage capsid because of error made during the life cycle of the virus. But then once the virus infects the bacteria, it will incorporate itself and it will take up the bacteria, it will eat it up. So the virus containing this gene then inject them into another bacterium, completing the transfer. There are two types of transduction. the generalized and the specialized type of transduction. You may read about it. The generalized transduction occurs during the lytic cycle of virulent and temperate phages, while the specialized or restricted transduction, the transduction particle carry only specific portion of the bacterial genome. And generally, the best studied example is the specialized transduction, which is the lambda phage type of virus. You can read more if you are interested. At this stage, you need to know that there are two types of transduction, the generalized and the specialized type of transduction. So look at this transduction of bacterial genes by viruses. You can see viruses in the environment. This is their capsid. And you have the DNA inside their capsid here. These viruses are just in the environment. But unfortunately, there are bacteria also around that environment. So this bacteria, for example, now is the host bacteria, is the host bacterial cell that will donate its own DNA to the virus. So this is the bacterial chromosome containing the gene, the DNA. So the phage is just moving around and it came in contact with a free bacteria and the phage injected DNA into the bacteria. Then what happened next? This is the phage. And the phage, the phage, that's the virus, will destroy, it will degrade the enzyme of the host DNA. After degrading it, there will be cell synthesis. New phage will incorporate the DNA of the bacteria that it has degraded. You can see this purple color now inside one of the capsids here. So transduction has taken place here. It is a mistake, but it has taken place. And then inside the virus, they multiplied. And then they are released into the environment. Again, any one of it that is released can also find another bacterial recipient, and the process continues like that. So that is how genes are transmitted between viruses and bacteria, and between bacteria and bacteria in the environment. Thank you very much. We are already in 12:45. Our next topic, I'm not sure whether we should leave it here and then do the final part on next week Friday, because I do not want to rush, but it is not a big deal. We are talking about Mendel's law of inheritance, which we have done even in our secondary school. Mendel's law of inheritance. We learned about Gregor Mendel, who is the father of genetics, because genetical work started from Gregor Mendel. He discovered 3 laws of inheritance: the law of dominance, the law of segregation, the law of independent assortment. Those are the 3 laws that guide how genetics functions. Mendel described the basic pattern of inheritance before the mechanism of inheritance. That is, before the gene was discovered, Mendel already carried out most of this work without knowing that it is the gene that is responsible for all these inheritances. Mendel conducted many experiments on the pea plants. Ogunbiyi Musibao, unmute yourself. You can speak. You are raising hand. Ogunbiyi Musibao. Okay, I go on. Mendel conducted many experiments on the pea plants. Mostly he used pea plants, Pisum sativum. And he carried out these experiments between 1856 and 1863. He studied the results of the experiments and deducted many observations. It was during his experiment that he came up with laws of inheritance. or Mendel's law of inheritance. It is important for you to understand the experiments performed by Mendel before learning the laws of inheritance. Mendel experiments on pea plants. Pea plants, how come it is pea plants that Mendel decided? He must have his reasons. Why can't he use another plant? Why pea plants? Pea plant was selected by Mendel for several reasons. For example, he observed that the pea plants easily increase in size, and it is also easy to preserve. He also noted that the pea plant has distinct characters that are easy to study or conceive. You can easily see the characters, you know, and also because this pea plant is available annually. He did not want to use plants that you can find during the rainy season and you cannot find during the dry season. So many of the generation of this plant can be studied within a short period of time. He also observed that this plant can pollinate itself naturally and can also be cross-pollinated. So the list of contracting characters which is studied in these pea plants is shown in this table. The plant is studied can have different flower colors. So it could be purple, it could be white. But you notice that the dominant color was purple, while the recessive color was white. You will soon know what is dominant and what is recessive, even though you have been taught in your secondary school. You also notice different plant heights, that some are tall, while some are short. He has different seed colors, the yellow color, the green color. And the seed shape could be round or could be wrinkled. And then the pod of the pea could be green or could be yellow. You also know that the pod shape could be inflated or may be constricted, that is flat. And the flower position could be axial or terminal. So these are the different characters, contracting character that Mendel had those 8 years or so that he studied. So he started by labeling the characters using different letters. For example, tall. So for a dominant tall character, he uses capital T. And for a recessive tall character, he uses a small letter T. Or Y, capital letter Y. If you see capital T and small t, that means that is heterozygous. It has a recessive gene for short, but phenotypically, that is the appearance, the plant appears to be tall, but there are recessive genes for shortness that may show up in the grandparents or in the grand upstream later. When you see something like YY, for example, if the color is yellow, that means the P is completely yellow because there's no small letter Y here. If it has Y, big letter Y and small letter Y, that means even though the P is appearing yellow, it also has some recessive color for white or any other recessive color. And if you see SS, short or small, whatever character is trying to store, SS, they are both small letters. So that means the character is recessive. So he used letters to represent them so as to be able to explain the genotypes for a particular trait. There are always 2 letters in the genotype. There are 3 possible genotypes: TT for complete tallness, TT for heterozygous, and TT for recessive, like I explained. The genes that are passed from the parent to the offspring exist in pair. These pairs are called alleles. So TT is an allele, is a pair. Dominant characters are described using capital letters and recessive using small letters. I have explained. So when 2 alleles are the same, e.g. TT or small letter T, they are called heterozygous, TT. Hetero means other, that is hybrid. I have explained earlier. Where the plant appears tall and has a recessive gene, which might express itself in the future generation, like I explained earlier. The way the plant looks is known as the phenotype. That is how the trait physically shows up in the organism. Like I explained, the organism might appear yellow, but if it has a small letter Y attached to the capital letter Y, that means it is heterozygous. And so that means it has a bit of another color which may show up in the later generation, maybe it's grandchildren or great-grandchildren. So it was on this basis of observation that Mendel proposed 3 laws known as the law of inheritance. And the first one is law of dominance. We should stop here and continue next week Friday, even though we need only 15 more minutes to complete this lecture. So it depends on you, but I will suggest whatever you want, you let me know. We can finish now.
Please continue, ma.
Since we are done with the lecture for this semester on my back. You are speaking for yourself, so please discuss among yourself. Let your class captain send me a message fast. I can spare you 15 or 20 more minutes to complete this. Though I have another presentation by 1:30, but I can do this. So let me know. I can do it now. I can also do it next week Friday. Which one do you want? I want Evans to tell me, please. I'm giving you 2 minutes to let me know your mind.
Let's continue, ma'am.
All right, thank you very much.
It's also here now. Ma, next week Friday.
Ma, let's finish it today. Just 15 minutes. Let's finish it today, ma.
All right, I want to assume that all of you want us to finish these lectures today. So
yes,
I will just continue in the next.
Ma, we don't say we want you to finish.
Yes, ma. No, ma. I don't finish it.
I don't understand though. Do you want it to
Yes,
be on next Friday? Eh?
No, today, ma. Let's finish it today, ma. Please,
Today,
ma.
ma. Today, ma.
Today, ma. Next week Friday, ma.
Friday.
Next week
Today, ma.
Today, ma.
Now that you have divided house, so
Next week Friday, ma. Oh, next week Friday.
We want it today, ma.
Want.
so see you next week Friday. Have a lovely day. Thank you. Though even though you guys, some of you guys disturbed today. Last week was better. It was nice, but today you were making noise. So we'll have it next week, 12 o'clock.
Okay, ma.
Thank you very much for coming. God bless
Okay,
you.
ma.
Thank you, ma. God bless you, ma.
God bless you.
Thank you, ma.
Yeah, you're welcome. All right.